Hi guys, welcome back to Bioinformatics. Now we're in week 6, and this week we're going to learn how to do PCR based cloning. I'm going to use an example where we take a domain from PLC gamma and clone it into an expression vector that makes a fusion protein that includes the green fluorescent protein. And the very top, here we are looking at the Course Angel website, and the very top file here is a paper published in Science in 2002 that describes this new vector. It's a very clever paper where they've taken the green fluorescent protein and they've altered a few amino acids so that it is now responsive to UV light. And when you shine UV light on the GFP, it becomes about 100 times brighter. So super cool. It's a neat vector. The bottom sequence here has the PA-GFP vector and also the PLC gamma vector sequence that we're going to use today. And then I also have a step-by-step example of PCR-based cloning here that will be similar to the lecture I'm going to give now on the iTunes, but it doesn't have sound, and it goes in a step-by-step lecture type fashion. So you may like that better, and you don't have to listen to my irritating voice. So it's up to you. There's also a homework at the end of that particular example. And then there's the Dropbox for the homework due next week on Friday, June 27th. Okay, let's go to Biology Workbench. Sorry, here we are. There, I've entered the two sequences myself. We're going to look at the vector first, the PR set PAGFP. And you can't tell anything from it here, of course, but we're going to copy. I'm going to copy and paste it into NebCutter and then analyze it for restriction sites, but it also shows us the different open reading frames. This is a plasmid, so normally it should be circular, but I did it linear because we're going to do some things with it. I happen to know that this sequence here is the 275 amino acid protein is the GFP. But when you're analyzing a new vector, I want to remind you that you can check yourself what all the open reading frames are by clicking on them and then running the sequence through NCBI and BLAST-P. So I'm going to do that quickly for that open reading frame just so you can be convinced that it is the green fluorescent protein. Okay, so now it's running. Here are the BLAST results. So you can see in the conserved domain database, it's found in GFP superfamily domain. Here are the alignments, and then here are the different protein, or the different vectors that were found, DNAs, proteins, etc. In this case, these are proteins. So the highest match was a yellow fluorescent protein, an enhanced green fluorescent protein. This eGFP is the one that they used in the paper, and then they modified it so that it was responsive. But you can see they're all different fluorescent proteins. So that is the proper open reading frame. And then we'll scroll down just for completeness so you can see the alignment here. And there's the protein sequences lined up by BLAST. Now back to NebCutter. Let's go back to the main display. We have a choice. Now if you notice, I'll zoom in here on the GFP protein, our open reading frame for the protein. There are a cluster of restriction sites towards the beginning of the protein on the left, and there's a cluster of restriction sites on the right. It is possible when doing cloning, if you're going to make a fusion protein, you can take your protein of interest, in this case the PLC gamma, and put it at the N terminus of your target protein. So that would be over here. Or you can also put it at the C terminus, which would be over here. How do you know which one to use? Well, the unsatisfying answer to that question is you don't until you try and see which one works the best. Sometimes if you clone at the N terminus, it disrupts the protein. So in that case, you would not be able to use it. Sometimes if you clone at the C terminus, it disrupts the protein. So you wouldn't be able to use it then. So in the lab, in real life, sometimes you clone in both places if possible, and see which one is expressed better, which one has the brighter fluorescence, which one is the best function, etc. You have to determine that empirically. For our example today, let's examine in more detail the N-terminus, which is here, and then also the C-terminus to look for appropriate restriction sites. So to do that, you have to keep zooming in. Now here we are zoomed in. This is the beginning of that gray box of the open reading frame. And so when I zoom in like this, I don't see any, right now at least, clear restriction sites. I'm going to zoom in so we can see the amino acid sequence. The nucleotide sequence and the actual start codon. So there's the amino acids, methionine, arginine, glycine, serine. And every time you touch one, you can see there's the ATG. That's the start codon. Arginine is CGG, glycine GGT, etc. Now we're looking for restriction sites. They need to be within the open reading frame for the protein. What I mean by that is if I clone out here my piece of protein from the PLC gamma, it is not going to be fused with the gene for GFP, because here's the start codon for GFP. Now I would have to have a start codon on my protein. They would have to be in frame with each other to make two proteins. That may work if you had a start codon, but we're not going to be using the beginning of the PLC gamma protein. So I have to embed my protein inside of the PLC or the GFP. When I look at some of these restriction enzymes that are over here, like NDE1, it cuts with one site, but it cuts right here. So I would be outside of the GFP protein, so that's not going to work. ACI1, that one also is, well, that it may work. It may not disrupt the start codon because it cuts after the G there. But notice that it cuts 45 times. That's no good. This is even worse. MSPJ1 cuts 624 times. That would just chew our DNA up into tiny little pieces. There's no way we would ever get it put back together again. 486. So it looks like, as I go through these, oh, here's 8 sites. That's still, we need one that cuts only one time. So the NDE1 would be perfect if we were using a start codon and we could get it in frame. So that's too bad. Let's look at the C-terminus, because I don't see anything right now that I would find really useful. I'm going to zoom in on the C-terminus of the GFP protein. There's a BSRG1 site. Inside of it, there's ECOR1, but it looks like it's after the stop codon. But let's zoom in more. Okay, we've zoomed in the BSRG1 site that I'd found before. It's right there. Okay, this is pretty good. It cuts one time, and it has the sequence TGTACA. And it is inside, it's close to the end, but still before the stop codon. Here's the C-terminus. That TAA is the stop codon for the protein. And so again, the BSRG1 is before that. That's good news. So that would be a usable restriction site. And in fact, that is the one that we're going to use. So we do have to sort of keep in our memory TGTACA, and then it cuts between the T and the G. If we look at some of these others, that cuts 4 times, 600. We saw that one before. There's the BSRG1 that we want. And so as I scan through, this is the only one that I find super interesting. Now, the other way to do this, of course, is to do a custom digest. You don't have to go through and look at each enzyme one by one. And then at the top of this custom digest where it says enzymes cutting N times, select that, put equal to one cut, and then you can look at all the enzymes that cut one time. You can select all of the enzymes, which I've done here, and then click on digest. And this will show you all the enzymes that cut your DNA once. And so you can see there are a series of enzymes at the beginning and the end that cut one time, including the NDE1 that cuts at the beginning. There's the NHE1 that cuts at position 134. That one may actually work too. Let's zoom in and see where that is. But that's another way to find these enzymes, the NHE1. So it's cutting in. We're getting a little, I don't know, it's this one would probably be okay. But notice we're about 10 or so amino acids into the protein. So I'd be worried that we would start interfering with the function of the protein. But I would probably in real life give that one a try in the lab. But our choice today is the BSRG1 site right here that's at the C terminus. So what do we do with this? Okay, well, now that we know the restriction site that we want, we go back to Biology Workbench. We need to analyze our DNA to make sure it doesn't have a BSRG1 site in an unusual place. So there's A miniata, the DNA that we want to use. Let's click on View. We can copy and paste this and bring it into NebCutter. Right now I'm just analyzing it for the BSRG1 site. I don't really, right now at this point, care about anything else. So we'll go to Custom Digest and look for BSRG1. Okay, it's not there, which is good news because we're going to build it in. Let's go back here and then look at the list of 0 cutters to confirm it. And there it is, BSRG1 does not cut our DNA. So that's good news because we're going to do PCR. We want to amplify a specific piece of DNA and we're going to build the restriction site into our primers. So back to Biology Workbench. Let me look here. This happens to be, let me copy this and go to the National Center for Biotechnology information. We're going to do a blast search on this PLC gamma protein, and I want to show you the different domains that it has. So here we are at the conserved domain database for the PLC gamma protein. You've seen this before after doing BLAST and in a variety of going through the protein database. It shows you all the little colored boxes are domains that were identified in our protein. And PLC gamma happens to have quite a few domains. The one that I'm interested in right now is this one called the SH2 domain. There's one, there's 2 of them actually in this protein. For this exercise, we're going to clone this N-terminal SH2 domain. And so to look at it more closely, you would click on the specific hit, the top one, and it would take you into the conserved domain database for that specific domain. So we're going to wait for that to show up here. I'm going to pause it. Here it is. It's shown up. This is the conserved domain database for the domain. Here, as you scroll down, are the alignments for the domain. The query is the one that we sent out. So this is our specific amino acid sequence starting at amino acid 532, HF, SEP, etc. And then it goes all the way down here to amino acid 634, and it ends up QCP, LRS, etc. So we need to find these amino acids on our protein and then identify the corresponding DNA sequence, and we'll do that in Biology Workbench. Select our sequence. There it is. To line up the amino acid sequence with the nucleotide sequence, we run a program called 6-frame. You guys remember doing this before. Now I happen to know that this is in frame 2, but if you didn't know, you would run either all 6 frames, or if you knew it was forward or reverse, you would run the appropriate frame. But I happen to know that my protein is made in frame 2, so I'm going to select that and click on Submit. And there is my protein sequence lined up with the DNA sequence. And I'm going to look for this particular amino acid sequence. And to find it specifically, you usually only need 3 or 4 amino acids. So mine is HF, SEP, WF. So I'm going to go back to Biology Workbench and search for that sequence. So you do Control F, HF. Oops. This one, notice they're 2 apart. So it's H, 2 spaces, F, 2 spaces, S, 2 spaces, E. There we go. So I've found the beginning of the SH2 domain. You guys see that there? So it starts at this nucleotide, or this codon, CAC, TTC, TCC, etc. I don't need to know the sequence actually, I just need to know the position. And so it begins at around 1,621 is that C here at the very end. So we have to count backwards. Let me do that quickly. So it turns out that this C right here is exactly 21 nucleotides back. So that's at 1,600. So we're going to begin our cloning at that position, 1,600. Now we need to find the C-terminal end of the SH2 domain. Whoops. So let's go back here. The sequence is QCPLRS. So let's find, QCPLRS. There it is. Oops. Sorry. RS. Whoops. Boy, I'm having trouble. QCPLRS. There, that's the end of it. So we started at 1600 and we're going to end up at this AGC, codes for that serine. So again, I'm going to count back to that serine. So that is 24 back to the S approximately. So that would bring us down to just under 1900. So just to make it easy in my head, we're going to clone the piece of DNA from nucleotide 1600 to nucleotide 1900. So we're going to clone a 300 base pair piece from the PLC gamma SH2 domain. So let's go back and talk about how to do that. We're going to use this software here that's called Primer 3. And let me quickly scan through here and find it. Let's see, there it is right there, Primer 3. This designs primers for the polymerase chain reaction. The polymerase chain reaction allows you to identify a specific part of your DNA. And using properties of DNA replication, you can copy an exact piece. So that's what we're going to do here. This is our sequence. Right now it doesn't really mean anything, but we're going to come down here and identify the region that we want to amplify. Our DNA is 3,816 nucleotides long. This second box allows us to identify the target region. If you remember, I want to clone from 1600 to 1900. So that's the target right now. Now they give you that you can identify 4 target regions, but we just have 1. Now down here we can exclude regions that we don't want the software to look for primers. Primers for PCR range in size from about 18 nucleotides to 27 nucleotides. And they'll cover an entire area, that whole entire area, 18 to 27. I usually like to use, at least for my first try, an area that's about 50 nucleotides, and that gives the program some space to work. So I don't make it just 18 nucleotides or 27 nucleotides. You'll see what I mean when we get the results. And so to get that, I take the lower number, 1600, and I subtract 50. And so that means we'll have in this excluded region from nucleotide number 1 to nucleotide 1,550. Then I do the same thing to this end, but I add 50 nucleotides. So we're going to start at 1950 and go all the way to the end of the DNA, which is 3816. You can see it up there. So what we have done is define the area where the software can look for primers. And so we're not going to let it look all the way to the very beginning or all the way to the end of the sequence. We want to force it to be close to our target region. We don't have a left primer or a right primer identified yet, so we're just going to skip that part. We're going to let it show us 5 primer pairs. The other thing that I often use to define my primers is the product size range. So this will do something a little bit different than having excluded regions. We're going to keep this one relatively simple, but I'll show you that it asks you for a low product size and a high product size. So how do you determine where to go there? Let's scroll back up to our information here. The product is the size of the piece that's amplified. And so right now we're identifying 1600 to 1900, so that's 300 base pairs. I want pretty much that whole sequence, right? We don't want to go too small, like I wouldn't want the primers to bind inside of my sequence and amplify only a tiny piece of that. So to restrict it to the ends, I'm going to make the low size 300. Then, if you remember, coming back up here, I subtracted 50 and added 50. And so the high size goes from 1550 to 1950, which is 400. And so we're going to restrict our amplification to somewhere between 300 and 400 base pairs. The rest of these parameters are details, and this is what makes the software valuable. In most cases, I don't change any of these because the default parameters are exactly what you need. But just to give you an idea of the things the software is used for, it calculates the melting temperature of the primers, so at what temperature the primers would bind to the DNA most efficiently, at what temperature would the primers come off the DNA. It calculates the GC content of the primers because you want them to be relatively similar to each other. It will also look within the primers for stretches of nucleotides that would be complementary to itself. So you don't want the primer to fold up and bind to itself instead of binding to your DNA. That would be bad. And so it looks for that. And how could that possibly happen? Well, if you have a 20 nucleotide primer, let's say the first 5 nucleotides were C's, so you have C, 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 and then the last 5 nucleotides were G's, G, 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 G. Then your primer may very well fold in half, and all the C's will bind to a G. So that's bad, and so it looks for stuff like that. Also, remember you have 2 primers, one that's in the going to bind to the 5' end of the DNA, and the other that's going to bind towards the 3' end. You don't want your 2 primers to bind to each other. So what if one primer had a bunch of C's and the other primer had a bunch of G's? Then they might bind to each other before binding to the DNA. So there's a lot of little things to look for, and that's what this software is doing for us to make the primers that are the best. The next section here we don't need. This is to look for oligo probes that you would use for doing library hybridization. And so we're going to skip all that and then click Submit. Now it's analyzing our sequence and looking for primers, and it's found some. Sometimes it won't, and you have to go back and change your parameters. So it reports what it considers to be the optimal primer pair or probe. In this case, we're looking for primers. There's 2 primers. The left primer, I'll show you in a moment, but binds towards the beginning of the sequence, which is we wanted, which was 1550. Okay, and the other one binds towards the 1950. It'll make a product that's 370 base pairs. That's shown down here. And then it gives us some of the parameters. The one primer is 58.5 degree melting temperature. It's 18 nucleotides long, has 50% GC content. This here, for any complementarity, it's looking for binding, and it's found some, but it's not high enough that it would matter. So what do I mean by that? For example, see these two C's here? And inside of the primer there's two G's in a row right there. So they may be complementary to each other. Or those two C's here and here that I had before, there's two G's in the other primer right there. And so 2 in a row or 3 in a row is usually acceptable. If you have 5 or 6 in a row, then you start to get into trouble. And so it's almost unavoidable. Like there's two G's in a row there, there's two C's in a row there. And so those are things that could actually affect your primer. But what it's counting on is all the rest of the sequence will help it bind to the DNA. Now let's look down at the DNA. So here we start at nucleotide number 1, and you see all the X's under the sequence. That goes from nucleotide number 1 all the way down to nucleotide 1550 right here. So that was the excluded region that we identified at the beginning of the program. And then we said, okay, starting here, you can start looking for good primers. And so it did, and then it found them. It found a good primer right here. So actually, those are those little arrows identify the primer sequence, which is up here. So that's our forward primer. The asterisks beginning here identify the piece of DNA we want to amplify. Notice that the primer begins before the sequence we want to identify. That's okay, because it looked for all these different parameters in the primer. Otherwise, you might be asking yourself, why not just start here and take the first 20 nucleotides and boom, there's your primer. You could actually try that, and sometimes people do that if you for sure want to have a specific site amplified. The problem there is that then that makes sometimes makes the primer less desirable, meaning that it'll bind to itself or it'll bind to other primers. It won't bind very well to your DNA. A variety of things. Its melting temperature might be too low or too high, so it's not useful in the PCR reaction. And so that's why we use the software. Same thing down at the bottom. The left-handed primer begins here and goes backwards. And it will amplify the DNA in between the two primers. And then you can see the X's again for our 3' excluded region. So there's our two primers. Now I do want you to notice one thing. The primer amplifies DNA, you remember back from our lecture on DNA replication, towards the 3' end. And so that's why the arrow is pointing backwards. The DNA that's going to be copied is the DNA between the two primers. So the reverse primer that I have highlighted in blue here is actually not this specific sequence that I'm showing you. It's the complementary strand of DNA that's not being shown. And so what do I mean by that? Well, this is the 5' end right here of this area on the complementary strand. And notice the sequence is G, T, G, and then C, C, C. C, actually there's 4. Well, let's keep that in your mind really for a second. G, T, G, C, 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 C. Now let's go back up to our primers. And if we look at the 5' end of that particular primer, here it is. And it's not GTGCCC, as a matter of fact. It is CAC GGG, which is the complementary sequence to the one we have down there. So that's the other strand of DNA going backwards. So that requires a little bit of mental agility. You have to think about that for a second and realize that the sequence of the primer will bind to your DNA. It is not your DNA for the right primer. But completely different story for the left primer. Notice this sequence. Here, we'll just take the first. This is actually relatively easy because it's doubled. CC, AA, TT. So cat, essentially. Now let's scroll down and see our primer sequence. There it is. It's CC, AA, TT. So it is the exact same sequence as the primer. So this primer, when you add it to the PCR reaction, would not be binding to this strand of DNA. It would actually, in real life, be binding to the opposite strand of DNA and copying, though, and making this particular sequence. So you have to remember that. DNA is double-stranded. When you make your primers, they have to be on opposite strands or bind to opposite strands of the DNA, which is what this software program is going to accomplish for you. Okay? Hope that makes sense to everybody. Now what do we do? Well, now we have our primer sequences. Also, here's the other four. It reported the four. And you might find these statistics interesting here. The software program on the left-hand side considered 1,834 different primers. Most of them are excluded because they're in the excluded region, so it gets rid of them. And then it starts getting rid of other ones. The melting temperature is too low, melting temperature is too high. There's 3' complementarity regions too high. And out of those 1,834, only 3 of them were acceptable. On the right-hand side, it considered 2,151, and only 7 of them were acceptable. And so then it starts to try to pair them up, and it considered 5 pairs and found all 5 to be able to work together. So I'm going to select the top 2. Those are the ones that we're going to use by clicking those little boxes. Zoom back to the bottom and import my primer sequences. So now we have our left and our right primer in our workbench, and we're going to have to do a couple other things to those primers. Okay, so this is the end of this first lecture because I don't want it to be too long. This is identifying primers. The next lecture is going to be on how do you use them to do cloning.